Okay, thank you. It is my pleasure to introduce our speaker tonight, Chris Stinson. Chris grew up in Rainy Lake outside of Fort Frances in Ontario, and he has been inseparable from all things animal related since he can remember. He moved to Vancouver in 1997 and completed an undergraduate degree at UBC. And he has been working at the Beaty Biodiversity Museum, I think, since about 2007, if I did my calculations right. He started as a volunteer, and now he is the lead curatorial assistant looking after the tetrapods. That's all things with 4 limbs, including mammals, birds, amphibians, and reptiles. On a more personal note, I wanted to say that throughout my time at the museum, I have always appreciated Chris's amenability to talking with visitors. So if someone would come into the museum and have a question that I was unable to answer, I would go outside the museum, find Chris. He would stop what he was doing and come out and spend some time talking to the visitors. So all in all, that really makes the visitor's visit much more memorable, and I really appreciate him being so flexible that way. His breadth of knowledge and experience extends beyond tetrapods to include things like insects, more animals, insects, marine invertebrates, and fishes, and participates in lots of field collections and bio blitzes that are done through the museum, pretty much all over BC, if I have my frame correct. In fact, Chris was one of the participants to Prince Edward Island and the bright red mud there with the excavation of our big blue whale, which is pretty exciting if anyone has not seen that, but Chris was very much involved with that expedition. He was also fully engaged in the research specimens getting into the cabinets as the Beaty Museum was starting to gear up for public opening. And he loves animals so much that he just doesn't mind at all dressing up as one. I just wanted to show this as one of the examples of not only his variety of knowledge or expanse of knowledge, talking about bats here, but this is Batman Chris himself. Chris is, I digressed a bit there. Chris is currently working on his Master's of Science through the Wildlife Coexistence Laboratory under Dr. Cole Burton of the UBC Forestry. He is here tonight to talk about his research on the Pacific and American Marten. Welcome, Chris.
Thanks so much for that introduction, Sheila. It's very nice. Good memories and nightmares about moving thousands of fish jars into the museum. But I threw out all my clothes when I was at the red mud in PEI after digging up that whale. But it was a great experience. Thank you to Nature Vancouver for having me talk tonight, and thank you for the scholarship that you guys gave me last year. And I'll just share my screen and get right into talking about some martens. Hopefully everybody can see that. So I'm going to be talking about Pacific and American martens, the taxonomic relationship between these 2 BC dwelling species. My work is determining the intergradation between the Pacific and American martens, how their ranges interact with each other, and exactly where their ranges are. And I'm doing this as a master's project while still working at the Beaty, perhaps somewhat ill-advisedly to do a master's while working full-time, but I'm doing it. COVID time. I'm talking about the Pacific and American marten and how their ranges interact in British Columbia. And like I said, I'm doing a master's while working full-time at the Beaty Museum. So that's an American marten in the background there. That one is from Ontario, where I grew up on Rainy Lake. Let me change the slides. Yeah. So here's my dad joke for tonight, the outline of a marten, to show my outline. I'm going to introduce martens, talk about the species of them that are found around the world, and then specifically about the martens in BC, and my study system and considerations like biogeography and historical context of migrating in. And since I'm doing most of this on museum specimens, I thought I'd show some museum specimen preparations and sampling. So there will be a trigger warning at one point where bugs are eating dead animals. I'll warn you again before that plays, but it will be on. And then I'm going to talk about the morphometrics of the caurina of the martens that I'm studying, and 3D imaging of the same cranium, the goals of my research, and then some other avenues of investigation. So what are martens? So they're in the weasel family, the Mustelidae. They're mesocarnivores, although some of them are omnivores. And I don't know if you can see my arrow, hopefully you can. They're this little beige section down here, and that's the Gulinae, the subfamily, which was just recently named, which includes the martens, fisher, wolverine, and the tyra. And this is jumping right into sort of a cladistic view of everything, trees, phylogenetic trees. I can't get enough of them. I love organizing museums, so that's where this comes from. And then this is on the right-hand side is the family that I'm talking about and their relationships, or at least one interpretation of their relationships. So to talk about the marten specifically, so those are in the genus Martes. There's 8 species, mostly in a circumboreal distribution with 2 subtropical and tropical species. So we've got the 2 in North America, American marten, Pacific marten, Japanese marten, beech marten, European pine marten. Quite often in Eastern North America, ours are also called pine marten, just to confuse everybody. The sable, which is in Siberia, yellow-throated marten, the biggest of the martens, and the Nilgiri marten, which is closely related to them. I'm going to go through briefly about them, tell a little anecdote about each one. And aren't they just so cute, even though they're vicious carnivores? European pine marten, they stole my salami when I was in the Cairngorms in northern Scotland, but I didn't even get to see them because I did it at 3 a.m. I guess I did put it out there for them. So they're in most of Europe, being reintroduced into England and Wales. They were trapped quite heavily. They've been, where they've been reintroduced, they're actually solving some of the problems that are eastern gray squirrels, which are also invasive here, but are invasive in the UK. Where they go, they wipe out the eastern gray squirrels and allow the native Eurasian red squirrels to rebound. Some of you may have heard of this marten when it shut down the Large Hadron Collider. This is the beech marten or stone marten. It chewed through electrical cables and shut the whole thing down for quite some time. It's found through Southern Europe and into Asia. And they figure it's about somewhere around $20 million it costs per year, because these guys like to chew the hoses and wires of cars and houses and things for some reason. Not sure why. They're one of the only martens that do so. But again, just chilling on a rock. Super cute little guys. This is a sable, so that's where these are from Siberia, as you can see in the map in the bottom corner. They're a bit bigger than our martens, but are closely related to them, and that's where you get your sable coats from. They're still trapped quite heavily. And if anyone's seen Happy People with Werner Herzog, there's quite a scene about trappers trapping them and smoking them out of trees and things like that. Now into the largest of the martens. It's the yellow-throated marten, found in Southeast Asia, up into India and Nepal. They're one of the few that have been documented to hunt in packs. Most martens are solitary, hunt smaller prey, but these will take down smaller deer and even gone after Himalayan tahr, which is kind of like mountain goat size. And this one here is hunted a macaque monkey, actually. If anyone's interested, you can actually see a series of photographs about that online if you're so interested and want to search that out. And then its close relative, the Nilgiri Marten from down in the southern India, in the Ghats down there, 6 small populations, and lots of sources put these as the same species as the yellow-throated marten, but they are quite disjunct, and they're in fairly small 6 populations down there. They weigh about the same. They're about the same size as the yellow-throated marten as well, so they get upwards of 4 kilos. They can get pretty big and formidable. Both of them are quite omnivorous, but they do hunt for meat as well. And then this cute little orange guy is the Japanese marten. You can see covers, this ranges mostly through Japan. The red on the map is where they've been introduced. Lots of them have this bright orange color, not all of them, but they're cute. They're a little bit smaller than our martens. And then how about the non-Martin Martens? These ones, I just coined the name Goulanids for Goulinet. I don't know if that'll fly, but we've got the wolverine, which most people are aware of, the circumpolar distribution, the largest of the terrestrial mustelids, take down a caribou or even a moose. The taira from Central and South America, quite large, but it's quite omnivorous as well. Eats a lot of fruit in its diet, depending on seasonality, but it's capable of taking down a deer, actually. They get quite large. And the fisher, a close relative of the martens in North America, doesn't fish at all. So that's an overview of the martens throughout the world and their closest relatives. So to get on to the 2 species that I'm studying are the Pacific marten and the American marten, both found in British Columbia. And within the last 10 to 15 years, Pacific marten was re-elevated to species from being lumped in with American marten because there was intergradation between them back when the biological species concept was the prevalent one. So I'm studying these 2. The Pacific marten goes from the coast of California up to Alaska, and on the coast of BC, including Haida Gwaii and Vancouver Island. American marten is found pretty much across Canada, from Newfoundland all the way over to BC, Yukon, and up into Alaska as well. So what am I studying? I'm studying these martens and what exactly is happening between them, the contact zone between the 2 species in BC, and how their ranges interact, overlap, and what specifically they are. You'll see with the conflicting maps that I'll be showing that this is yet to be established and is a area of contention between various mesocarnivorologists. And I'm also looking specifically at the morphology of this, what's happening in the zone between them morphologically with their skulls particularly, but perhaps with their pelage and things like that as well. So why is this important? Well, taxonomy is important, especially for people that work in museums. But it's the species are a basic biological unit, as most of you already know. And in order to understand life, we have to understand what's out there. And it has important implications for conservation and management of harvested species, which martens are. They're probably the most trapped species in British Columbia still, and it's still a fairly lucrative market for their furs. So determining which species needs what habitat and that sort of thing is quite important. And in order to do that, you have to define what the species are. Taxonomy also is a building block for understanding other branches of biology like evolution, ecology, genetics, as well as issues of today like climate change. We can look back at the history of of a particular species and figure out where it migrated and things like that. And taxonomy is the basis for that. And then most importantly to many of you, as well as me, is it's crucial for everyone's life lists so that they can keep track of the proper names for everything and organize their museums properly as well. Got to find those things in drawers. So getting on to defining species, I won't go through all of those that are on the right-hand side there, but there's all sorts of species definitions. I'm inclined to follow the general lineage concept, which basically means that there's a separately evolving lineage, and then you can use the factors from any of the other species concepts as evidence towards that, rather than simply defining it as something that interbreeds with fertile offspring, as in the biological species concept. I think that's more holistic and allows you to build on things rather than push things out. So it's the basis of what I want to do is help define species with just a little bit more information on this small system that I'm working on. So the system I'm working on is British Columbia, and we have a complicated biogeography history in BC. We have all the mountains and we had the glaciation. It's a bit more complicated in British Columbia because of that, from the last glaciation than it was in Eastern North America, where it just receded and you had basically the same flora and fauna increasing to the north. We had all sorts of migration routes like this on the right. So both from the north, because Beringia was ice-free during the last glacial maximum, and we had lots of small ice-free areas on the coast, Haida Gwaii, Brooks Peninsula, for instance, and then down towards the south, like Eastern North America did. So it's very complicated, and BC is sort of a crossroads of biogeography. So you have lots of eastern species meeting up in with their western counterparts that have been separated for tens of thousands of years in various parts of BC. For instance, like birds meeting up in northeastern BC. So you have boreal coming across from the east and meeting in the Peace region with like a wren or something from the southwestern of the continent, and they meet up there, and that's their contact zone between these two species. So then we get winter wren and Pacific wren. And martens fall into this same thing, although their contact zone is not up in northeastern BC. It's a little bit more hard for mammals to migrate through the mountains than it is for birds to fly over them. So American marten was in eastern refugia and managed to migrate to and invade most of North America. And the Pacific marten was restricted to the small refugia on the coast and down in either the coastal refugia further south or the southwestern refugia and move north. So which is why we get such complicated interactions between ranges of species in BC and my particular study species, the American marten and Pacific marten. So this was only a few years ago that this map came out of American marten. This was before they did the split. And as you can see, marten are found pretty much across BC. Any of the gaps you see is probably lack of sampling or trapping in that area rather than them not being present. The exception being some of the central Okanagan where there aren't that many trees, but even there they do pass through. And then on the right, we have a map that was made in about 2013 that sort of highlights a possible hybrid zone between the Pacific marten in the pink and the American marten in the green. And I want to figure out if this is right and make it more detailed and get more information from it. So this is an older map on the left here of all the subspecies of American marten at that time. This little scalloping line is thought to be a different version of where the contact zone is. with its black line, where Vulpina starts as the southern Rockies range for Pacific Marten, and everything to the east and north of that is American Marten. But it's unclear whether this is correct. More recently, in 2018, it's a paper by Jocelyn Colella has shown that it's really unknown in British Columbia where the actual contact zone is between these. And as you'll see in the next page, there's some thought that American Marten comes all the way down even to the lower mainland, although this is probably from a lack of information rather than a true representation of the range of American Marten, because you have them overlapping in this crosshatched area where, between the green and the blue. But somehow they haven't come up here because they used to be in the Cascades, so it's unclear why there wouldn't be some sort of overlap there. And this study on the left-hand side is from northern Idaho, southeastern BC. And as you can see, the caurina on that side don't even make it up into British Columbia. So it's unclear how, from this study, there are Pacific Marten up further north of here connecting this. How did they manage to get through a gauntlet of American Marten? So as you can see, it's a bit confusing on how either Marten's ranges overlap, where they are exactly. You'd think that we would know this kind of thing, but we don't. And this is the beetles that I was talking about. So what I'm working on, rather than doing field work, I'm working in the museum, and part of my crew to collaborate with my research is the dermestid beetle colonies. You can see mostly larvae here, which help me clean up a whole bunch of skulls. I've cleaned upwards of 1,000 so far. Well, the beetles have. I've washed them after they're done. And this is just a little scale of the skulls as the beetles clean them up. And the next page will be, if you don't like squirmy bugs, look away, because this is a time lapse of the beetles eating the flesh off of the skull. So this was overnight when they were particularly voracious that I took that. And as you can see, they like to move the skull around while they're eating it. And I don't know if you caught it, but at the bottom, the hyoid process, so little bones of the throat of the marten, actually got trucked away to the left-hand side. I also am doing preparation of skins. So most of my specimens are from trappers, so they don't have skins, but a lot of them are also salvaged from roadkills or other accidental killings of martens. I don't actually collect them directly. So I also prepare them. There's the inside out skins up at the top. I'm taking pictures of the inside out ones to see if there's any sort of pelage difference on the inside pigment of the skin. You can see that it matches up with their throat patches and things. And then I get a round museum study skin like this. And we have historical ones that I'm hoping to put into my data set as well. So as I was saying, lots of museum specimens. At the Beaty, we were up to over 2,000 from BC. And then we have a whole bunch of opportunities to get loans from other ones. Because of COVID, I've sort of put the focus just on the Beaty thing, so I don't have to worry about shipping during this time. And I was hoping to go visit more local museums at some point, but that'll have to wait until after COVID is over. Hopefully I can get some good data from just the Beaty Museum specimens that I've been collecting over the past while. And we won't have to worry about too many loans or traveling to other places. I'm also looking at archaeological specimens, which I'll mention later. So I'm working on cranial morphometrics, but how else do these species differ? So what I have here is across the top, some females, and across the bottom, male skulls. On the left-hand side are ones from Saskatchewan, so you can see they're a bit longer, a bit narrower across the zygomatic arch. And then we have Vancouver Island are the middle ones. They've got a shorter face, wider, and then on the right-hand side are from Haida. So these are a bit of the extremes of the range, just to give you an idea of it. I know they just look like skulls, but all look the same, but there is quite a bit of difference between them. The Haida ones are the biggest of them. There's also quite a bit of difference in body size, but this is complicated by the island forms being a lot bigger and there being a large overlap between the sizes of them. And the Pacific marten that's found on the mainland ends up being quite a bit smaller than both their island counterparts of Pacific marten and the American marten that has come in from the east. There's been some thought about using pelage to tell the difference between them, but this is more from an individual basis because you could camera trap them and figure out what they are because there's just too much individual variation. There seems to be a bit more orange in the throat patterns of Pacific marten, but it is really hard to quantify. Obviously, genetics would be good to do. I'm hoping to collaborate on that after my master's, focusing on morphometrics. And then there's some evidence coming out of ecology, behavior, and diet being quite a bit different. So American marten has been more studied because it's found in Eastern North America. And they tend to have large home ranges and disperse really far when they're, especially the males. But especially the island forms of Pacific marten, Haida, Vancouver Island, and maybe the coast as well. They have smaller ranges. They don't have to go as far for food, and they end up not being as curious or not nearly as arboreal as well. It seems that the ones in Haida don't even, from a study by David Burrow a few years ago, they won't even climb a tree to go for food. There's too many dead deer around in Haida and fish all around and ground nesting birds. So they don't bother climbing trees, even though somebody decided to bring red squirrels to Haida to feed them at one point. So to further talk about my morphometrics, I'm going to use traditional morphometrics, so measuring with calipers and geometric landmark measurements as well, making use of more advanced technology with computer generated 3D images using photogrammetry. So that's where you take a bunch of photos and stick them together to make a 3D image. To begin with, though, I'm working on 40 cranial measurements on each of these little skulls, but I'm only doing a subset after I take the photogrammetry to sort of check to see which one is giving more information. And then I'm also going to compare it to the two-dimensional geometric morphometrics. That's a mouthful. But first, I will be using the 3D imaging analyses. So you can test on shape, measure curves. It's a lot more powerful than just a straight up set of calipers. So hopefully it'll get more information from them and I'll be able to more finely define what each species is by their skull shape. I'm using a photogrammetric software called Agisoft Metashape, which is actually made in Russia, but it's one of the few that works on Mac and is also quite cost effective, especially for educational institutions. So I have a setup, and this is where I'd like to thank Nature Vancouver, because this is what the scholarship money went towards. That light box and turntable that's there was purchased with that scholarship money, and thank you very much for that. So I take 3 different angles. I don't actually change the angle of the camera, I change the angle of the skull, and then do a 360 with an overlap of a third, so it ends up being about every 10 degrees, so 36 photos at each different angle for the skull to reconstruct a 3D image, and then do multivariate analysis on the shapes and the differences between them using the 3D images. So this was my first attempt at just a 360-degree image. It gives you an idea of what I'm going to do. There's been some glitches with the software, so I wasn't able, and the delays of COVID, I wasn't able to get a model up and running. But here is what I'm hoping to do, so this gives you an idea, that's 48 photos all the way around. And then I can analyze those through the software. It creates a 3D image that you can spin and measure in all sorts of ways. And I'll be using landmarks as well as targets on there so everything is scalable and you can get fine details to measure all the differences between the skulls. So what I want to end up doing is defining that range. And as I said, this is an older map of and it was thought to be this sort of scallop thing here, but new data from genetics and things suggest that maybe American Marten is either encroaching on this even more, or we were just wrong to begin with. So we'll see what happens with my morphometric analyses. And I'm also hoping to combine this with ecological demographic data that was all collected for my specimens as well. Melissa Todd at Nanaimo Ministry of Forest, Lands and Natural Resource Operations and Rural Development has been doing gut analyses, doing aging, and testing for diseases in all of the martens that I've gotten from them. So it should be interesting to combine all that data to help figure out where the two species meet in BC and what's happening between them, and hopefully later on do some genetic work as well. I also mentioned that I wanted to do some paleontology. Martens don't fossilize particularly often, being forest species, not conducive to fossilization except in caves. So this is a diagram here of the Charlie Lake Cave up by Fort St. John. That's had quite a few marten fossils in it, and I'm in contact with the archaeological collections at SFU, Jonathan Driver there, and he's hoping to do some ancient DNA techniques on that, as well as allowing me to do some morphometrics on it. As you can see, all we get is some fragmentary bones, so it's a bit more difficult to do analysis with this. But there are a few fossils that I'd like to include in my work to help give a paleontological historical context to how the martens have changed in their ranges over the years. So like I said, my goals will be to figure out that big question mark in BC as to where the two species meet, what's happening in that meeting zone, and then going from there, perhaps developing some conservation efforts and things that will help them survive. Down in the southern ranges of like the Humboldt Marten, the Pacific Marten down here, they're quite endangered, and there's only, I think, 11 or something left that they've detected on the Olympic Peninsula. So as we develop more in BC, that could be a problem for the Pacific Marten as well. American Marten seems to be doing quite fine. They have such a massive range that it's probably not an issue, but the Pacific Marten, also coupled with their different ecological niche and smaller ranges and smaller dispersion rates, might be a problem. I'm also hoping that I can discern something from the traditional morphometrics and maybe tease something out that will allow field techniques for easy identification between the two. I'm hoping for that anyway. And then a pathway to further studies on ecology and genetics. And also, I'm hoping to create a methods paper comparing the 3D, 2D geometric and traditional morphometrics on a low budget, rather than getting these $30,000 amazing laser scanners. I want to do it sort of that anybody could just go to the store and buy a camera and do this sort of analysis and compare the two together. There's another Marten from Ontario in that little corner. That little guy tried to get a red squirrel on my deck, and he was upset because he was being scolded by it, and then having me follow him for like 20 minutes. They have interesting vocalizations, Martens do, grunts and growls and squeaks and whirls, all sorts of things. In the future, I was hoping to maybe do some microscopic hair structure analysis where you can do casts of hair in resin to figure out what their scales look like. And there might be a difference between them. They haven't been apart that long, the two species, so it may not be the best idea. Also spectral analysis of the pelage. There's some mustelids that fluoresce under UV light, so there may be something in that as well. And I've always been fascinated with the way that mammals can tell each other apart and tell so much stuff by smell. It would be interesting to hook up a camera trap that you could have an electronic nose on that would allow you to tell an individual or tell a species by a simple chemical test of the air. But that's sort of a pipe dream way down the road. And then as I develop these specimens that I already have and figure out if there's any gaps, perhaps I can do some actual targeted field sampling, more likely from trappers than directive sampling, budgetary concerns and things like that. And then collaborations of studies in genetics after my master's is over. Just want to thank everyone here at Nature Vancouver for the scholarship that allows me to do this research. My collaborators at the Government of British Columbia, Dr. Cole Burton at UBC, and all my colleagues at the Beaty Museum, including Ildiko is here, and Sheila, and I saw several other people as well. This is some Haida Marten specimens that I brought back in coolers. You should have seen the look on the guy, the X-ray technician at the airport, trying to figure out what was in all of these cases that I sent through on my way down from Haida. So this is just a summary. I won't read through it all again, but I just want to figure out what's going on in BC between these two species of mesocarnivores, these martens, and using museum specimens to do it with some 3D imaging techniques. Thank you very much.